we're going to explore first the idea of molecular diffusion. And once we get a basic understanding of that, move on to think about how we go from molecular diffusion to synthetic biological behaviors. So let's jump in and see what's going on and what we can make with it. The photo here is of a frog egg, a frog. And it's just sitting there. It hasn't turned into a frog yet. And I want you to think about these questions. Like, what is underneath how life works? Why does a frog egg turn into a frog? Why is it happening? So it turns out this image is from a movie, a historical film, an incredible film of frog development. So let's take a look and see what's happening. Again, we're starting here, looking just at a frog egg, ready to go. Here we go. We see the cell divide, it looks like, into two. Why did that end in 4 cells? Wait a minute. Why is this happening? Let's go back in time. It was just sitting here. We have one big cell. And suddenly it's starting to change and do things. I don't see anything coming in from the outside, like cutting and dividing and making all of this happen. It's all happening from the inside out. Something underneath and inside this living matter is causing all of these changes and behaviors, these cell division events. Oh, look at what's happened now. Just keeps going. Why is this happening? We fast forward, we start to see forms and shapes appear, starting to begin to look like the plan of a body plan of an animal. And eventually we get tadpoles. And eventually a frog. But remember where all this started, right? It's just, we had a cell and it started to spontaneously divide and then differentiate and turn into a tadpole and then a frog. We know that cells are comprised of biomolecules, and we know that life happens in water. So the molecules within a cell are mostly suspended in water. Of course, there's other things in there too, but for now, let's just think of it as biomolecules suspended in water. On the left is an image of a simple, quote unquote, simple microorganism, not a frog. And on the right, we've seen this before, is a beautiful representation, a representation of how the molecules might be organized in this cell. The green in the bottom shows us the outer membrane or shell of the cell. And then in purple, we see, I don't know if those are ribosomes for making proteins. And then maybe in yellow, we've got DNA or RNA. Not quite sure, but we could figure it out. Oh no, it looks like maybe the RNA is in this pinker color and the yellow must be the DNA. It's so interesting. Once you can see things, you can begin, what would it be? What could it be? Now these images, they're static. They don't answer the question of like, how did that egg turn into a frog? Let's keep going. Now, in keeping it going, I also want to help us and challenge us to develop a deeper empathy for what it's like to be one of these molecules, what it's like to be a cell. So the tool I like to use is a magic wand, and it's a certain type of magic wand. It's a magic wand that lets us make things bigger. And we can use it in this case. Cells are tiny, so let's use this magic wand to make a cell a lot bigger, just so we can help think about it. And when we start to imagine things moving around, hopefully we'll be more comfortable with what might be going on. So I've listed in the middle here different types of molecules or objects we can find in cells. A water molecule, two hydrogens and one oxygen, or a base pair of DNA, an A and a T base pairing in this case. GFP is a green fluorescent protein. Maybe our cell has a little protein that's green when you excite it with a wavelength of light. Bilayer, this is lipids that make a membrane that could encapsulate the cell or regions within the cell. The ribosome, a combination of molecules that when you give it messenger RNA and other inputs, it can make proteins. A phage. Phage are only in cells if they're infected. A phage is a type of virus for bacteria. The full name for phage would be bacteriophage, which means eater of bacteria. mRNA, messenger RNA, that's going to instruct the ribosome what protein to make. The genome, the DNA template that when transcribed makes the RNA that then gets okay, so these aren't all the types of molecules you'll find in a cell, but some ones that we'll talk about again and you should know about. Cells are tiny, so if we use an example cell, say E. coli, the bacterium that lives inside the human gut, how big are these molecules? So E. coli roughly is 1/1,000,000th of a meter long. Sometimes it's a little bit bigger, and it's shaped, like a tube or a cylinder, about 1 micron or 1/1,000,000th of a meter long. And that's notated by 1e-6. This means shorthand for 1 × 10⁻⁶ meters, or 1/1,000,000th of a meter. Okay, so how big is a water molecule? How big are all these other molecules? And you can say water molecule is about 2.8 × 10⁻⁻⁻ of a meter. A nanometer would be 1 × 10⁻⁹. So this is not even a nanometer. It's 28% of a nanometer, it looks like. How about a base pair of DNA? 3.4 × 10⁻⁻10 meters. What about this green fluorescent protein? Well, it's about 3 nanometers, or 30 tenths of a nanometer. I'm keeping everything in 1 × 10⁻⁻10 just so you can compare it more easily. Protein's about, this protein, typical protein, maybe 3 nanometers in diameter. Bilayer, 4 nanometers, or 40 tenths of a nanometer. A ribosome looks like it's approximately 20 nanometers. And the phage, 580 tenths of a nanometer, or 58 nanometers. mRNA, 3,000 tenths of a nanometer. The genome, 1.6 millimeters. I jumped there, right? Genome's like big. Looks like it's going back. This is incredible to think about. It looks like a genome in a bacterium like E. coli is so long, it's going back and forth over 1,000 times just to fit within the cell. Now it's very skinny, but wow, it's just going back and forth, back and forth. And it's not necessarily organized literally like a cable, but it's like, Now, of course, these things are moving, and this is what our current discussion will end up being about. So this GFP, we can represent how quickly it's moving, and this will make more sense in a few slides, but roughly it's moving about 6 microns, or 6 × 10⁻⁹ meters per second. Excuse me, 6 × 10⁻⁶ microns per second. A micron is 1/1,000,000th of a meter. All right, so see what I mean? Like, cells are tiny. These numbers, they're real or approximately real. And by the way, there's a great site on the internet called BioNumbers where you can get quick access to a lot of numbers like this. Will be useful in your back of the envelope calculations as a bioengineer. But unless you've already been operating at the scale of angstroms and nanometers in your life, it's hard to have empathy or make sense of this. So that's why I like this magic wand. So what we're going to do is we're going to take E. coli and we're going to say, boom, 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 it's 100 million times bigger. We're going to multiply all the dimensions by a factor of 10 to the 8th, or 100 million. Not going to go billion, that'd be too big, but let's see what happens when we go with 100 million. So we're going to multiply all these dimensions by 100 million. And we're just making a bigger cell. We're not adding more atoms, but just like magically everything's bigger so we can see what it feels like. Now, when I make something 100 million times bigger, and originally it was a micron, now it's about 100 meters long. Like multiply 1 times 10 to the minus 6 times 1 times 10 to the 8th, you get 100. So now we have our magical cell, and it's about 100 meters long, which conveniently is about the size of the bioengineering building at Stanford, which is called the Shriram Center for Bioengineering and Chemical Engineering. And so we're going to call this magically enlarged cell Escherichia Shriram, E Shriram. Suddenly, it's like a lot easier. I don't know about you, but it's going to be a lot easier for me to think about, what's going on inside this object. So a water molecule is suddenly 2.8 centimeters. Well, that feels nice. It's like an inch. And a base pair of DNA, it's suddenly much more accessible. It's like 3.4 centimeters, roughly. And this green fluorescent protein that was so tiny before, 3 nanometers, 30 centimeters. What is that? Like the size of a basketball or football, approximately? The bilayer encapsulating things, 40 centimeters, almost half a meter. The ribosome that's making proteins, it's 2 meters tall. It's about 6 feet tall. That feels friendly. Like I can deal with that. The phage, this virus that be infecting a cell, is kind of intimidating. It's 6 meters. Wow. Interesting to think about that. It's like a 2-story building. The mRNA that's being translated to make a protein, it's about 100 feet long. It's like a little string 30 meters long. The genome, encoding all the DNA and stuff, 160 kilometers. And if you, not that it looks this way, but if you packed it up in the building and like had it be like really densely configured, back and forth, 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 1,600 times, the cross section of that bundle would be about 4 meters square, right? So that's kind of interesting. Like it fits. It all fits within this 100 meter building. Now, remember, things are moving, right? I mean, something is causing life to happen. That frog embryo develops. The ribosome makes proteins. For the ribosome to make protein, it either has to move along messenger RNA or spool and pull the messenger RNA through it. And we know that it's running at about 50 nucleotides or letters on the RNA per second, depending on conditions, but like roughly that fast. And so we can do the math, like how long is the base pair? And if we're transiting, 50 bases a second or nucleotides a second, and each base is this long, oh yeah, like that ribosome is moving at about 1.7 meters per second. That's a brisk walk, right? It's not a sprinter. It's not like the 100-meter dash, right? You do 100-meter dash in 10 seconds, if you're fast, right? And so like a sprinter is running at 10 meters a second, maybe a little bit faster than that, depending how great you are. So ribosomes like walking along, fast walk. This green fluorescent protein, this example protein that's the size of a football, soccer ball, basketball. Yeah, it's moving at, looks like 650 meters a second. I don't know how you think about that. For me, that's just like, whoa. Remember, the whole cell, size of a building, is only 100 meters. And I've got proteins in this object, size of basketballs, and they are zooming around at almost a kilometer a second. And I think we'll have to figure this out. This is actually not like the instantaneous velocity, as we'll see, but this is the average displacement over time. So in the super microscopic, it might actually appear to be going even faster. Okay, but hopefully, I really invite you to explore and walk around Escherichia Shriram, the cell the size of a building, because suddenly you can get a sense of like the relative sizes and how things are moving roughly, and how does it all fit together, right? So fun to think about the E Shriram. So we've seen this video before. I want to reuse it to go inside this building and remind us of a couple things, but also come back to the question, like, what's underlying life happening? This is a computer simulation of molecules comprising cytoplasm of a bacteria. Like most of the molecules people know about. And the molecules, they're all biomolecules, have different colors depending on what they are. And you can see they're just jostling around. They're moving around seemingly randomly. Like, what's causing them to move? They have little like rocket jets. No, that we don't see any evidence of rocket jets. Or is somebody coming in there with a stick and like poke, 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 lots of sticks poking this around? Not from the outside, at least. What's not shown here is what these molecules are suspended within. Are they in a vacuum of space? No, they're inside a cell. And we know on our planet, right, life operates mostly within water, almost exclusively. And so the invisible thing not shown here are the water molecules. And so we know this jostling is happening within a watery milieu. We'll come back and look at this video again once we understand diffusion a little bit better. Remember what happens if you freeze a living system? It won't be living, right? If the ice crystals disrupt the structures, then it's broken forever. But yeah, like things moving in water, that movement is essential for life as we know it. And so that's interesting, right? We could change how quickly things are moving by changing the temperature. In 1827, there's an important brief account. Look at the title of this. We've talked about reading research papers. Here's one from 1827. A brief account of microscopical observations made in the months of June, July, and August of 1827 of the particles contained in the pollen of plants, and on the general existence of active molecules in inorganic and organic bodies by Robert Brown. This is a really important research paper, to say the least. And Mr. Brown, among other things, was apparently interested in life. And if you looked at objects derived from living systems, like the pollen of plants, he could observe that grains of pollen suspended in liquid just jostle around, are moving around by themselves, seemingly randomly. Maybe this capacity to move around spontaneously is the secret essence underlying life, that there is some life essence within living systems that causes this apparent spontaneous movement. My understanding is one of the experiments he tried was to take grains of pollen and observe their motion, as shown here, and then to take some of those samples and cook them, basically, autoclave them, sterilize them, so that the grains of pollen would still be there, but they'd be dead. So that any life force or essence within them that was causing this spontaneous movement should go away, because they'd be killed, right? But they'd still be these physical grains of pollen, they'd just be killed. Interestingly enough, when you do that, they still move. Something else is causing the pollen grains to move about. Brown's work is going to be developed over time into what is recognized as Brownian diffusion, or spontaneous motion of objects suspended in water or another solution. Here are two representations of this, again, very simple computer simulations in 2 dimensions in this case. And I encourage you to look at the links below if you want to go deep on this. But for an introductory course, just want to get a sense of what's going on. So first, let's look at the movie on the left. We are going to have particles moving around. They're moving around randomly. And some of the particles have been colored yellow. And the yellow particles, we're going to follow their traces over time to see how they're moving. And of course, the particles might run into each other, and they'll be moving around. So let's just see what happens. They're moving around pretty fast. And the traces shown in blue are kind of all over the place. It's just, I don't know, I describe it as random. Now, of course, if we wanted to claim they were random, we would have to really carefully validate the statistics of the trajectories. But it looks random to me. It's really fast. The movie on the right is a little bit different. It's exactly the same as the left, except one of the particles suspended in solution is big, this big yellow particle right in the middle, at least to start. And so let's run that movie. The first thing you notice is the particle that's larger is moving more slowly. The trace isn't very long, so it's, it looks like it's going up in this case mostly, but it's like drifting around, so it might still be random. I'd want to follow it for a longer period of time to see where it ends up, and does it go in all directions. But it's being jostled around by the smaller particles and other things that might be invisible. And so this is a way of thinking about what Brown was observing, those pollen grains moving around. And so how is this going to be related and more simply organized into what we know today as Brownian motion, Brownian diffusion? The first thing we can draw upon, beyond looking at what's happening at a microscopic level, is to just think about what happens. Imagine you had a perfectly smooth dish of water, and you were able to put a drop of food coloring in the middle of the dish. And you didn't disturb the water at all. And there was no flow in the water. And you're just looking over time to see how the dye and the food coloring, well, what would it do? What would you expect it to do? In my experience, when I've done things like this, there might be a concentrated spot of dye in the middle of the dish right when I add it, but inevitably, for reasons, the dye will spread out over time. And so that's what's shown in this depiction on the bottom left of this slide. Like the height of the surface shown in this image is the density of the dye in a two-dimensional solution. And as time progresses, the intensity of the dye diminishes as the dye overall spreads out and colors the entire dish. If I wanted to think about that instead of a two-dimensional system, just something on a string or on a line in one dimensions, I might have a peak of something that I add at an early time, and that peak spreads out as things diffuse over time. And what's interesting here is suddenly we just went from a microscopic representation of how things are spontaneously moving around to a bulk or aggregate representation, which means when you have many molecules interacting as a system, things behave as an ensemble set of molecules or in bulk, right? And the bulk representations are often not dominated by noisy behavior. Instead, we see this very smooth, almost determined behavior that follows our expectations. Our expectations, again, are derived from empirical observation. Like I add a drop of food coloring to a dish in perfectly still water, and the dye will diffuse or disseminate and become less intense over time. Diffusing particles, we know from experience, tend to spread out over time. Diffusing particles moving around spontaneously. It turns out mathematics can describe how far molecules move due to this spontaneous Brownian motion, named after Mr. Brown. And we're going to use some math here. And it might look a little strange at first, but it's not too complicated. So really encourage you to, if you haven't seen it before, just play with it. Such beautiful math. There are three equations here. From top to bottom, it represents how far molecules move due to spontaneous random motion in a one-dimensional system at the top, in a two-dimensional system at the bottom, in the middle, and a three-dimensional system at the bottom. What do I mean by 1, 2, 3D? Imagine you had a molecule on a strand of DNA or a strand of RNA, and it was sitting there, and it could just like randomly go back and forth. Like how far would it tend to go over time? And the equation at the top is going to help us think about that. Instead, let's say I had a membrane on the cell and a molecule like sitting inside the membrane or outside the membrane. It was just like randomly searching around the membrane. Like how far would it tend to go over time? We could use the middle equation. Or let's say I have a molecule inside the cell in the middle of cytoplasm. Obviously, it's a three-dimensional space it can explore, and we can use the bottom equation for that. What do the terms mean in these equations? X squared or R squared is known as the mean square displacement. the square of the average distance traveled by a particle in one of these systems. So the left side of the equations is the square of how far something is going to move on average. The letter D here on the right side of the equation is the diffusion coefficient. It's the oomph. It's like how fast is something going to be moving, we think. And T is the elapsed time. So to state this in simple English, if I want to know on average, how far something's likely to travel, I am going to take the diffusion coefficient for the something, for the molecule, and the amount of time I'm willing to let go, the elapsed time, and I multiply those two terms. And if I take the square root of the product, following these equations, I'll get the value to x or r, and that'll be my mean square displacement, the average distance traveled by a molecule with that diffusion coefficient and that amount of elapsed time in the system. Let's do an example. Remember green fluorescent protein. It's a protein a couple nanometers across, and it's going to exist in cytoplasm if we have the DNA encoding that protein in the cell and it gets expressed. We can look up the apparent diffusion coefficient, the value of D for this protein in typical bacterial cytoplasm in this case. And to keep it simple, the value of D, the diffusion coefficient, is 7 microns squared per second. So if we wanted to ask, well, how far will green fluorescent protein tend to move on average over some period of time, we can take this value of D and decide what system we're working with, like a 1D, 2D, or 3D system, and plug it into the equation. So let's do it for a cytoplasm. All right, so we use the 3D equation, and we get the number 6 times the diffusion coefficient of 7 microns squared per second. And let's just ask in a second, how far will green fluorescent protein go? And so we just add 1 second in. And so we're just filling in the bottom equation with our values. And we see that the mean square displacement is 42 microns squared, which is a little bit strange. We want to turn this into just microns, or millionths of a meter. And so we need to take the square root of 42, which is about 6.5 microns. A protein of the size of like green fluorescent protein, relatively typical protein in cytoplasm, everything else being equal for bacteria at room temperature, maybe body temperature in a second, it'll go about 6.5 microns. That's pretty interesting. Remember, the cell, the E. coli, is about a micron. So like this thing's moving, right? It's moving around. The fact that molecules are moving spontaneously because they're being jostled by the underlying invisible movement of the water molecules is what's going to enable living systems to behave, to be alive, to spontaneously perform, and in the case of the frog embryo, start to undergo development. What's really interesting when you start to adopt this mathematical thinking is you can ask questions, like, oh, let's say I wanted to go twice as far. How much longer would that take? You'd be like, well, if I want to go twice as far, I need twice as much time. Right? Look at that 3D equation, any of the equations. To get the average distance traveled in units of length, you actually have to take the square root of the right side. So if you want to go twice as far, you actually have to wait 4 times as long, because you're going to take the square root of t. All right, so that's one of the powers of moving this thinking into some mathematical representation. Let's unpack the diffusion coefficient. And once we get through this, we're going to pause, catch our breath. But I want to unpack the diffusion coefficient just to give you deeper intuition for what factors are going to influence how molecules are moving around. And so at first blush, you might have never seen this before. It might be intimidating, but it's not too complicated. So let's just play with this diffusion coefficient equation. So D is the diffusion coefficient for a molecule. Each molecule could have a different diffusion coefficient. We'll see in this example why there could be differences. The numerator has 2 terms. The first thing is called the Boltzmann constant, which if you haven't heard about is going to be esoteric. The second thing I bet you've heard about is just temperature of the system. And so right there we can see, if temperature goes up, diffusion coefficient is going to go up. because temperature's in the numerator. Boltzmann constant's a constant. And you'll be asked in the PSET to make sure you have a feeling for what it means. But in simple language, the Boltzmann constant takes temperature and lets you relate temperature to the velocity or kinetic energy of molecules, right? So it's like, how fast is something going to be moving? The temperature is the energy of the system, but how does that energy of the system translate to molecular motion? That's what the Boltzmann constant is going to let us do. Okay, so we've talked about the numerator. Again, the Boltzmann constant isn't going to change. The temperature could change. If the temperature changes, that's going to impact the diffusion coefficient. In the denominator, we have 6. Okay, that's a number. We have pi, that's a constant, another number. Then we have eta. We have viscosity. Viscosity is like how thick the fluid is. Think of syrup for a pancake. It's like really thick. If we have a thick liquid, it's going to be harder for molecules to move around. So as viscosity goes up, the denominator increases, diffusivity is going to go down. That makes sense. That's viscosity. And then R is the radius of the molecule. Like how big is the molecule? And the bigger the molecule, well, it's going to be in the denominator. So the denominator goes up and diffusion coefficient goes down. It's interesting. The only thing that's specific to the molecule in this example of the diffusion coefficient is the size of the molecule. Right. And then the system is defined by the viscosity of the liquid or the solvent and the temperature of the system. Right. So those are the variables. And just to summarize, diffusion follows temperature, meaning if temperature goes up, diffusion goes up. Temperature goes down, diffusion goes down. If temperature goes all the way down, right, there's no diffusion, so you're not going to have a living system. So it's going to be static, right? It's going to be frozen. Although technically frozen is a little bit, okay, you get the idea. The inverse of viscosity, so if viscosity goes up, because it's in the denominator again, diffusion is going to go down and vice versa. And diffusion follows also the inverse of molecular radius. The molecule gets bigger, diffusion is going to get slower. A molecule that was 4 times smaller should diffuse about twice as far. Because remember, when we go to the preceding slide, to get the mean square displacement, distance traveled on average, you have to go through a square root. Right. So hopefully this gives you, some framework to chew on and think about what's going to impact how fast molecules are moving. I want to recommend to you, if you love reading books, this incredible book, Random Walks in Biology by Howard Berg. And the link here will take you to a website where you can actually find, I believe for free, the chapters of this book. But if you can't get it, let me know and we'll get you the chapters as best we can. It's such a beautiful book about how randomness and spontaneous behavior, bottom up in biology, manifest as beautiful biological phenomena within living systems. Okay. So now we're going to return to something we're familiar with, this representation of molecules jostling around in cytoplasm. And what I want to ask you to do is just pick a molecule. We're going to let this video loop, right? Like let's pick, look at this little red molecule down here. Find the tiniest molecule and just follow it around. Is what we're observing consistent with the framework we've just developed for representing spontaneous diffusion of molecules, Brownian diffusion? Look at a tiny molecule like this tiny red molecule. It seems to be, what would I expect? It should be moving more quickly relative to the larger molecules, and that looks to be true. Where's a big molecule? Like this molecule over here on the right, it's like a Christmas molecule. It's red and green. It's not moving as quickly. Its diffusion coefficient must be lower. That makes sense. Another thing I want you to think about is whether a molecule, is it only stuck in one neighborhood or subregion of this, or do the molecules eventually get to go everywhere? And this is trickier to see because at first blush, when I look at this video, it seems like things are really locally constrained. But if you pay closer attention, there doesn't seem to be any formal constraint keeping things in the top left versus the bottom right. Instead, over time, when it's easier to see with the smaller molecules, they're able to go everywhere. This is what's called a self-mixing system, such that over time, on average, anything can be anywhere. This idea of self-mixing, that on average anything can be anywhere over time, for at least some of the molecules inside living systems, is going to make the engineering really different. It's not like, oh, I've got a transistor and another transistor and I've got a wire and I connect the two and they're stuck there. Of course, we are going to be able to connect some things in biology to other things, but underneath it all is this self-mixing foundation. And for a lot of things, like the business of life requires that everything can move around and be everywhere else on average and interact and, sample or intertouch everything else, right? Very interesting. So spend enough time with this, find the video online if you like, and just, all these videos, make sure you're just like gaining empathy for what's going on with the molecules inside living systems. So we're going to wrap up for now and offer these reflective questions. Which molecules move faster or slower? Why? Do things make sense to you? Given enough time, how many other molecules will any given molecule interact with? In a diffusing system, will all molecules eventually interact, i.e. collide with everything else? Thinking ahead, how are we going to get organized behavior from this underlying randomness or Brownian motion? Stated differently, what's good about this underlying spontaneous self-mixing from an engineering perspective, and what's going to be a challenge? See if you can guess how that might end up.